Okay, here we go. I'd like to give a report on this major paper that came out in Nature. We've talked about a little bit online. I'm going to present two views of this paper. I'm doing my best to follow in the steps of my idols to be a good scientist, and that sometimes, most of the time, is a difficult, painful path, and not everyone wants to come along on that path. So I'm going to present sort of an easy take and then a second, more difficult take. Number one, I'm very happy to read this paper. It was really fun to read, and it's a great paper. Number two, it is a quintessential example and a superb example of translational research. Is in that third category between basic science and clinical research. This is a great example of translational research. Number three, it is filled with exciting potential leads for and clues for targets for future molecular therapies. And lastly, yes, it deserves to be published in Nature. Okay, so now close the door, and anyone who's not interested in a more difficult take, please turn off your computer now. It deserves to be published in Nature. It definitely deserves to be published in Nature, but not because it's a great science, but because it represents millions of dollars in multiple myeloma research foundations, money, donors, and contributors, a tremendous amount of work. The reason why it's in Nature, just in case anyone out there is curious, is because of the this. This is the supplementary data, which you should print out and see. It's much larger than the paper itself, and contains a phenomenal amount of data. So it deserves to be published in Nature because so much time and effort has gone into this, and patients have donated their samples for this. So it should be published for that reason. Third point, filled with new clues. If we knew this was a list of definite not targets, that would actually be helpful. The problem with science that isn't very good is we don't know. It's damning with faint praise. Filled with possible targets means we don't really know yet whether these targets are any good, so it really doesn't really help us very much. In the supplementary data, there's hundreds and hundreds of different mutations and targets listed. Certainly in there may be something wonderful. The problem is we just don't know, and so we need to do more work. And I must say, kudos to Chapman and the authors for being humble and calling it an initial analysis, because that's clearly what it is just the tip of the iceberg. Point number two, outstanding translational research. But if you read the blog at all, I don't believe there really is such a thing as translational research. There's basic science research, there's medical research, clinical research, and then there's marketing. So this is high profile because it's potentially very exciting for patients and for medical research. It's not medical research, and it doesn't really provide us any insights into the basic science either. So it's a great example of a type of research that as a whole I don't really believe is very good. And lastly, am I happy to read it? Yes, I'm really happy to read it. The main reason is because we're working on a similar study, full disclosure, and yet so I was terrified that they would do such a good job that it would leave no room for our experiments. But I'm very happy to read it because it was done in such a way that there's clearly room for improvement. And specifically the technical issues that I'm concerned about. Number one, they use blood as their normal control blood cells, and blood cells we know are contaminated with circulating myeloma cells. They also have memory B cells, which share mutation profiling with the myeloma cells. So the sensitivity and specificity of mutation detection is going to be hampered by that, by the selection of controls. Number two is they didn't screen the patients. They took all the patients that they could, samples that they get their hands on. So that means multiply retreated and relapsed patients. So the number of mutations and the type of mutations, it's good to generate a lot of data, but this list that they have is filled with things that are probably red herrings based on the lack of patient selection. And lastly, that they didn't do any screening. So we know they didn't screen their samples for genetic and genomic stability. We know these genomes are highly unstable, highly aneuploid, and so mapping the sequencing reads in these cases is extraordinarily difficult, and again hurt the sensitivity and specificity because they apparently took all comers. If you're interested in more technical stuff, I think Dan Cobalt on his blog Mass Genomics is probably going to read this paper and provide an analysis from a more technical standpoint. So stay tuned, and thank you for your feedback. Over now.